To test for starch, we use a brown liquid called iodine. We add a few drops to the food sample in a spotting tile, and if starch is present, iodine will turn blue or black. If there's no starch in the sample, the iodine will remain brown. To test for protein, we use a blue reagent called Biuret. We add a few drops of blue Biuret solution, and if a food contains protein, it will turn purple. If not, it will remain blue. To test for sugars such as glucose, we use a blue solution called Benedict. We add a few drops of Benedict to the sample in a test tube, then place it in a water bath at around 95 degrees. The colour change that occurs depends on the amount of sugar in the sample. A positive result with a small amount of sugar would be the sample turning green or yellow, whereas samples with a lot of sugar will turn orange or even brick red. With a negative result for sugar, the Benedict remains blue. To test for lipids or fat, we use ethanol, which is a colourless liquid, and water. We add a few drops of ethanol to the sample in a test tube, add water, and shake. A positive result for lipids is a white, cloudy or fatty layer forming, known as an emulsion. With a negative result, no emulsion forms. You may also use a reagent called Sudan 3 or Sudan 4 or Sudan red. This still forms a fatty emulsion, but it has a red colour. These are qualitative tests. This means they can tell us whether each nutrient is present, but they can't tell us exactly how much. They're also quite subjective because they rely on detecting a colour change, which can be influenced by people's opinions. Here are some practice questions for you to try. Pause the video and give them a go. To test for protein, we use Biuret. To test for starch, we use iodine. To test for lipids, we use ethanol. And to test for sugar, we use Benedict. A positive result for protein would be a colour change from blue to purple. For starch, it would be a colour change from brown to blue-black. For lipids, we see a cloudy emulsion forming. And for sugar, we see a colour change from blue to either green, yellow, orange or red, depending on the amount of sugar present in the sample. To test a sample for sugar, we add a few drops of Benedict to the sample in a test tube. We then place the test tube in a water bath at around 95 degrees and observe the colour change.